Khatib series in Human Anatomy and Physiology. Chapter 2. Cell Structure and Function. Episode 6. Cell Transport. Part 1. Passive Transport. In this session, we'll be diving into the essential topic of cell transport mechanisms across the plasma membrane. In general, cell transport mechanisms across the plasma membrane are essential for maintaining homeostasis, allowing cells to acquire nutrients, expel waste, and regulate their internal environment. These processes are divided into two categories: the passive transport and active transport. Each of these mechanisms plays a unique role in cellular homeostasis depending on the type of molecule being transported and whether energy is needed or not. During passive transport, molecules move down their concentration gradient from an area of higher concentration to an area of lower concentration. Also, passive transport occurs also without requiring energy, or ATP. On the other hand, you can see the active transport as the movement of solutes against their concentration gradient form an area of low concentration to a higher concentration. Also, note that this process requires ATP as energy source to accomplish the transport of molecules. Nonetheless, the process of passive transport includes three key mechanisms, including simple diffusion, facilitated diffusion, and osmosis. Let's start with the first type of passive diffusion. That is the simple diffusion. This is the movement of small nonpolar molecules like gases across the hydrophobic lipid bilayer of the plasma membrane from their high to low concentration. Since these molecules are small and nonpolar, they can freely pass through the membrane without any assistance from any transport proteins. Examples of this is the movement of oxygen and carbon dioxide between the lung and the blood during the process of gas exchange. As per this image, oxygen moves from the alveoli where its concentration is high into the bloodstream, where its concentration is low. On the opposite, carbon dioxide moves in the opposite direction from the blood into the lungs, following its concentration gradient. Now, let's move on to facilitated diffusion. Unlike simple diffusion, facilitated diffusion occurs for larger or polar molecules that cannot easily cross the hydrophobic lipid core of the plasma membrane on their own. In this case, the molecules move down their concentration gradient from high to low concentration with no need for energy but with the help of transport proteins. This transporter could be a channel protein or a carrier protein. An example of facilitated diffusion that uses a carrier protein is the transport of glucose into the cells. Because glucose is too large to pass through the lipid bilayer by itself, it relies on a glucose carrier protein called glucose transporter or GLUT. On the other hand, we also have ions channels that transport ions in and out the cells like sodium and potassium ion channels. Ion channels function as external and internal doors, allowing specific ions to enter or exit the cell, thus regulating cellular functions. The external and internal doors are also called activation and inactivation doors, respectively. When a channel is at rest, its external door is closed and the internal door is open, preventing ion flow. Upon activation, the external door opens to allow ions to pass. After activation, the channels is inactivated or locked by closing the internal door to prevent further ion movement and overactivation. Ion channels come in various types, each with a unique function. First, let's talk about ligand-gated ion channels. These channels open or close in response to the binding of a chemical signal known as a ligand with a receptor on cell membrane. Examples of ligands are neurotransmitter or hormone. When a neurotransmitter like acetylcholine binds to the receptors on a muscle cell, it opens sodium channels, allowing sodium ions to enter and triggering depolarization, which leads to muscle contraction. Next, we have voltage-gated ion channels. These channels open or close in response to changes in the membrane potential or the voltage across the cell membrane. Voltage-gated ion channels are especially common in excitable cells like muscles and nerves. A great example is the sodium and potassium voltage-gated channels. When the cell membrane depolarizes and becomes less negative, channels open, allowing sodium ions to flow in, which rapidly triggers an action potential. Then, we have mechanically gated ion channels. As the name suggests, these channels open or close in response to mechanical forces, such as pressure, stretch, or changes in cell shape. These mechanical forces cause physical deformation of the membrane, which then opens the channels. Mechanically gated channels are critical in processes like touch, hearing, and proprioception, allowing the body to respond to external stimuli like sound waves or pressure. Examples of some receptors that work by mechanically gated ion channels are baroreceptors, which detect the changes in blood pressure, Merkel cells in the skin, which detect pressure and light touch, and hair receptors in the cochlea of the ear, which are crucial for hearing. Finally, we have the leak channels, also known as background or passive channels. These channels are unique because they are not gated by any external signal like voltage changes or ligand binding. Instead, they are randomly gated, meaning they periodically open and close based on an intrinsic rate or random fluctuations. While leak channels are not fully open all the time, they allow ions such as potassium and sodium to passively diffuse through the membrane, following their concentration gradients. This continuous movement of ions helps establish the resting membrane potential in excitable cells, like neurons and muscle cells. The last type of passive transport is osmosis. Osmosis is the movement of water molecules across a semi-permeable membrane from an area of higher water content to an area of lower water content. As shown in this image, osmosis can occur freely and directly through the lipid bilayer by simple diffusion following its concentration gradient. However, this process is generally slow due to the water's relatively small size and polarity. To enhance the movement of water, especially in cells where rapid transport is necessary, water channel proteins called aquaporins are employed. These aquaporins act like gates, allowing water molecules to pass through the membrane more efficiently and quicker than simple diffusion while preventing the passage of other molecules or ions. They are essential in cells in areas like the kidneys and large intestine, where water needs to be reabsorbed efficiently back into the bloodstream to prevent dehydration. This process is also regulated by hormones. For example, the hormone vasopressin, also known as antidiuretic hormone, rapidly activates signaling cascade to increase the expression and insertion of the aquaporins in collecting ducts of the kidneys and intestinal cells to increase water absorption. However, the direction of water movement during osmosis is regulated by the concentration of the solutes in the solvent. To understand this, look at this image. You can observe a semi-permeable membrane separating the right and left sides of the container. Also, the right side of the container is more concentrated, containing more solutes. However, the left side is diluted, containing less solutes and more water. This means that water content is more in the left side of the container as it contains less solutes. Herein, water will follow its concentration gradient and moves from the left side to the right side of the container to balance solute concentration. Therefore, we conclude that osmosis is driven by the concentration gradient of solutes, meaning water naturally flows from areas of low solute concentration to areas with a higher concentration of solutes, such as salts or proteins, in order to balance concentrations on both sides of the membrane. From our previous discussion and the relationship between solute concentration and water movement, we conclude that movement of water or osmosis is determined by tonicity. Tonicity refers to the concentration of solutes in a solution, relative to the concentration inside the cell. The relationship between osmosis and tonicity is vital because the type of solution surrounding the cells will determine whether water moves into the cell, out of the cell, or remains balanced. By understanding osmosis and tonicity, we can predict how water behaves in different environments and how cells will respond to changes in their surroundings. As shown in this image, a red blood cell is floating in a liquid environment. In this case, the solution surrounding the cells is an isotonic solution. This means that the concentration of solutes outside the cell is the same as inside the cell. Therefore, water entering and leaving the cell is balanced and the cell stays in equilibrium. A good example for this situation is when you're sick and receiving an intravenous normal saline solution in a hospital which is normally isotonic. This normal saline won't cause your cells to shrink or swell, keeping you in a stable state with healthy cells. Now, let's place the red blood cell in a hypotonic solution as shown here. You can observe that the concentration of solutes is lower outside the cell than inside it. This means that water content outside the cell is higher. As a result, water moves into the cell to balance the solute concentrations inside. Therefore, influx of water causes the cell to swell, and if too much water enters, the cell can burst in a process known as hemolysis. A good example of this is water intoxication that occurs when you drink excessive amounts of water over a short period of time. This dilutes your plasma and lowers blood sodium levels in a condition known as hyponatremia. As a result, blood plasma has higher water content which enters the cells leading to swelling and hemolysis. In our third example, the red blood cell is placed in a hypertonic solution where solute concentration outside the cell is higher. This makes water content inside the cell is higher than outside. In this case, water moves out of the cell to balance the solute concentration outside, causing the cell to shrink and shrivel. This is what happens in several situations such as drinking seawater, excessive sweating or diarrhea where your plasma become concentrated as you gain sodium or losses water. This creates a hypertonic environment in your blood and hypernatremia, which can lead to cellular dehydration, organ failure, and damage to tissues.